So I would like to welcome everyone to this edition of New Frontiers in Spatial Omics. So today is October 28, 2024. So just as a benchmark for new technologies this time next year. So I'm Peggy Angel. I'm an associate professor in the Department of Pharmacology and Immunology at the Medical University of South Carolina. Today we are joined by a guest chair, Colleen Crouch. So Colleen is assistant professor in the Department of Mechanical, Aerospace, and Biomedical Engineering at the University of Tennessee, Knoxville. Colleen, do you want to tell us briefly about your laboratory research?
I would. And hello, everybody. So the Crouch Imaging Lab uses preclinical ultrasound imaging to study cardiovascular biomechanics in various diseases and physiological states. And why I'm here today is we also use mass spectrometry imaging to study spatial distribution of lipids in cardiovascular tissue. And we also have some brain studies.
Thank you, Colleen. So today's topic is multimodal spatial analysis and improved identification for proteomics. So a current topic in spatial analysis of the proteome is leveraging the ability to combine imaging proteomic information with modalities that are used to measure biophysical properties of the tissue microenvironment. There's also an ongoing challenge with being able to improve our proteomic identifications directly from tissue. There's many isobaric peptides that may be seen in tissue imaging, and we want to be able to separate those apart. So a main protein that we're going to talk about today is collagen. So these contribute greatly to tissue biophysical properties. And they share homologous regions between proteins. They're especially challenging in identification. And so, Colleen, do you want to highlight our agenda for today?
Yeah, so for today, we will have two presentations from our three speakers, followed by a fireside chat with the presenters that will hopefully include audience discussion. So we really want to facilitate a discussion between speakers and our audience.
Thank you. So our first speaker today is Professor Martina Marchetti-Deschmann, who's full professor at TU Wien and heads the Institute of Chemical Technologies and Analytics. So she obtained her PhD in chemistry from the University of Vienna, Austria. Her research focuses on mass spectrometry with an emphasis on multi-instrument and multimodal imaging. She is recognized internationally for innovative methodologies in combining molecular and elemental imaging, contributing significantly to the field since 2003. She has published over 147 peer-reviewed articles, holds 4 patents, and has received several prestigious awards for her work in mass spectrometry. Martina?
Oh, thank you, Peggy, for the introduction. I'm going to share my screen. Here we go. I hope you can see can you confirm that you can see my screen, please? Wonderful, thank you. So thank you for giving me the opportunity to introduce our work and our expertise to this audience. I developed from the field of mass spectrometry, but recently I started to connect to other scientists at my university, and we have a very strong material science faculty. And together with them, we try to understand biomechanics on the tissue level, and we want to understand that also on the molecular level. And that means that we have to start to combine different methods which were not combined before for biomaterials. We are working on completely different scales, which makes it rather challenging. And that is why we do it. We want to gather comprehensive information. So what are we doing? Where do we come from? So material in general has heterogeneity, especially in the biological field. And the heterogeneity depends on the composition of the material of the tissue, and of course also on the chemistry. This is more relevant for material scientists, but in the end, when we want to find out why a tissue is stiffer or more elastic, we really need to know what is the underlying composition in terms of proteins, maybe lipids or small molecules. Tissues and biomaterials can be described in different ways, which are usually terms you face in material sciences. That's hyperelasticity, viscoelasticity, poroelastic characters, and a hierarchical character. So viscoelastic behavior means that a material can be explained by entanglement and disentanglement processes on a molecular level. So in the end, either it is rather strongly cross-linked or not. So you can understand that that might play a crucial role, especially for collagen and collagen fibers. And poroelastic behavior is the term used to describe the integration between fluid flow and solids deformation with a porous material. And if this sounds a little bit too strange, a little bit too material science, like you can imagine cytoplasm of living cells behave as a poroelastic material. So you see there's a little bit of softness given compared, for instance, to a cell membrane. And hierarchical material, we're not talking about that today. That's typical bone structures. And when we are talking about biomaterials, we all know they consist of those different types of molecules, proteins, peptides, lipids, metabolites, salts, and don't forget about water content, which is crucial on how soft or how stiff certain material can be. So what are we now looking at in terms of size of such material? I think you all know that from bone to eukaryotic cells, we are overspanning a large dimension scale, and not all of these biomaterials have the same stiffness. And an expression term for elastic stiffness is the elastic modulus, as you see here as the title for this graph. Everybody knows steel, and we know that steel is not very soft. So if we have a high elastic modulus in the gigapascal range, we are talking about very, very hard material. And if we move to the right on the skin, you see bone. It is not as hard as steel. I think we all know that. They can break. But we are also, for bone, multiple orders less elastic compared to articular cartilage, to muscle, or to the eukaryotic cell. And this elasticity, this softness, this stiffness of a material can be tested, but we also need different methods for this testing because a steel block is much larger than a single cell. So we have to consider that we are working in the range of centimeters to the molecular level, which is in the nanometer scale. So how are we actually measuring the stiffness of a material? The scientific term for that is that we are testing the stiffness or the softness of material by indentation. Indentation means we press a object, as you see here in gray, the triangle, a hard object with a very well characterized material stiffness or softness onto a surface. We press into the surface, meaning we are loading the tip. The tip has a certain indentation, so we are going into the material with a certain height, and then we are moving the tip away from the surface, from the material, and we are measuring how quickly is that indentation going back to its original form, or if it's going back to its original form in principle. So for steel, you can imagine this will be an indentation. You will have a triangular scratch or tip in the surface. But for the soft tissue, of course, this is going back to more or less its original form. And Exactly these two steps are very well measured with a so-called AFM tip. This is a tip which is on, and you will see how that looks later in the scheme, is mounted on the stick, let's say like that, and then you can really measure how deep, and this can be nanometer levels, this can press into the material. Measuring the pressure you need to press into the material is called the loading, which is coming up to a certain pressure you need to press into the material for a very well-defined depth. And then when you are releasing, unloading the material again, you can measure again the load on the surface of this tip, and you get a second curve. Mathematically, when you are taking the steepness, or if you are calculating here actually the steepness of this curve, you can define the stiffness or the softness of a material. For biological materials, these in the beginning used tips showed to be not adequate enough, and that plays a role for the example I will give you. If you take a triangular shaped hard tip and you load the tip with pressure, the strengths or the forces are distributed radially into the material. And if you press further, it still goes radially, which is, of course, not a very well model for a biological system. And over time, it was found out that actually spherical tips pressing into the biological surface much better represent the forces the neighboring cells, the neighboring tissue material is seeing, and this is much better describing better describing the measured pressure, which is much better reflecting the stiffness of a material. So in the end, for biological materials, at least we here in Vienna, we are using spherically, very often non-geometrically similar indenters to measure the stiffness of the surface. And what is the indenter? The indenter is actually the object pressing into the surface. This is for biomechanics, the way how you can check for elasticity, for viscosity, for poroelasticity. In the end, these physical-chemical properties strongly relate to chemical structures and chemical compositions, and the mass spectrometry today allows you to measure molecules unbiased. So basically, you can measure unknowns. And by identifying the mass to charge ratio, when you're coming from mass spectrometry, you know that a molecule needs to be ionized. Then you can measure in the mass analyzer the mass to charge ratios. And from that, you can actually calculate back the molecular mass of the analyte. And mass spectrometry allows you to give information about particles. You can measure molecules, you can measure elements. And by this, the more accurate you measure these mass to charge ratios, you can even determine chemical structures. And if you're running a fragmentation experiment, so you are choosing one of the mass to charge ratios, you intentionally collide it with an inert gas, you induce fragmentation, and from these fragments, you can even further nail down structure. For peptides and proteins, that means you can deduce amino acid sequences, and by that identifying potentially identifying proteins. Biomechanics and molecular information can, of course, be collected from bulk material, but in the end, when we are looking into tissue, we also want to know the order and the ordering and the localization in the tissue. So what we want to do is actually to know the localization of those molecules. That means we are moving to mass spec imaging, where we are collecting one, or where we are selecting one mass to charge ratio, either from a fragment or most commonly from the intact molecule. And we draw pictures. Basically, we recalculate the localization of a certain mass to charge ratio in the tissue. And as imaging is much easier understood than the mass spectrum, we also try to bring that back to the biomechanics. So we now try to draw images from the elasticity within a biological tissue, so the indentation information, in correlation to mass spectrometry molecular information. And how we do that, I will now present you on one of our main objects of interest. We want to understand how a meniscus starts to tear, or what is actually going on in the collagen material when we are tearing our menisci. So as you know, the meniscus, or as you may know, the meniscus is a disc-shaped piece of cartilage. So it consists mostly of collagen proteins of different collagen types. In the cross section, it is the meniscus has a triangular shape, so it's getting thinner towards the inner part of the knee and is rather large at the end. It is connected to the bone material, to the collagen fibers, and meniscus, what is also very important, does not show the same blood flow throughout the whole material. It is less, it shows less blood, or a lower number of blood vessels on the inner side compared to the outer side. What also has to be mentioned for meniscus is that the collagen, which is forming fibers, has a radial and a circular orientation in the meniscus, meaning that which makes actually the meniscus so flexible towards movement, towards torsion, and keeps our knees intact. Nevertheless, over times, and especially when you're getting older, your meniscus tends to tear, not only because you are maybe too active, but also just due to aging. So it is also important for the medical doctors to understand where and how tears in the knee, in the meniscus occurs. You know, you can have a tear, which is a lateral tear in the meniscus, or you can have a tear from the side. This is the critical one, because this tends to get larger and larger and larger, and then your meniscus starts to tear totally. So you're having actually two parts. How is the collagen looking like in such a meniscal material? Collagen is a protein. Peggy was mentioning that, but this protein, which consists of amino acids, is three-dimensional structured in so-called tropocollagen fibers. So you see we have a helical structure, which is wrapping around itself. This 300 nanometer scale tells us that we have now already an object which can be measured, maybe even by indentation. So that means we can look into the stiffness or into the hardness of such a collagen fiber, which means if the meniscus is still elastic and is still flexible, we have a lower number of potential meniscus tears compared to collagen, which is strongly maybe cross-linked and by this much stiffer. The dropper collagen, this 300 nanometer dropper collagen molecule is forming collagen fibrils. These fibrils have a diameter of approximately 1 micrometer, and multiple fibrils are forming together a collagen fiber. And what is tearing is the collagen fiber. So when you have a meniscus tear, you are tearing apart more than just one proper collagen formation. Peggy mentioned that already. There are multiple collagen types. In meniscus, the so far best described collagen types are collagen 1, collagen 2, collagen 3, and to some extent collagen 5, 14, and 17. These are the main collagen types, but of course all the other collagens are also available, but to a significantly lower extent. So what is known from the meniscus in terms of biomechanics? So that's not an unresearched field. What you see here is a scheme of a meniscus. You see the smaller inner part, the broader, the wider outer part. You see that we have here we have different colors. This comes from the more blood holding outer part of the meniscus, which is also called red zone, because it is a lot of blood in there. Then we have the inner part, which is called the white zone, with only a very low number of blood flow, let's put it this way. And we have the mixture with this, the white region of moderate blood vessel. There have already been nano indentation done, and you see here there's this small spherical object, which is the intender, which is pressing into the meniscus. And what they found is that we have a rather high modulus on the outer part of the meniscus in the red zone, and which is significantly different from what we see on the inner part of the meniscus in the white zone. And you see here the just an excerpt of the publication the colleagues presented. You see that even if you are looking into the horizontal versus the vertical fibers in the meniscus, we also see a difference in the elasticity. Of course, for the vertical fibers, so if you are pressing from the top of the fiber onto the meniscus, onto the collagen fiber, we have a significantly different behavior for the collagen fibers, while when we are looking at the horizontal parts, it is staying more or less the same. So this was our starting point, and what we wanted to know is what is the underlying molecular information. So we needed to understand what what microindentation, what biomechanic behavior is occurring at a certain region of interest in the meniscus, and what does this region of interest hold in terms of molecular information, which we tried to identify by mass spectrometry imaging. So what we want to understand is the correlation of mechanics with composition. So for the nano indentation experiments, we went for a meniscus, which we received from patient materials. We cut the tissue into tissue sections of different thickness, And these sections are mounted on glass slides. And here plays the thickness a crucial role, because if you consider that we are pressing onto this tissue with an intender, you can understand that the stiffness of the sample holder, the glass, might play a significant role, especially for very thin tissue sections. So what we found out is that we had to go actually to rather thick tissue sections. This is a thickness which is usually not used in mass spec imaging. We are working with 5 micrometer thick section in the standard mass spec imaging experiment. But for a 5 micrometer thick section, you are drying the section to mount it, and you mount it on the slide, you dry it. That means it deflates even further, and we would more or less measure the stiffness of the glass, which we do not want to have. So we went for a thicker section, we dried it, and we are now measuring actually only the biomaterial. Unfortunately, this image looks rather blurry, but this is how the meniscus looks like. So exactly like in the scheme, we have a triangular shape with the red-red zone, the red-white zone, and the white-white zone. Having mounted our tissue on the glass slide, we of course wanted to understand the biomechanics under physiological conditions. It doesn't help if we have a super dry tissue section, because this is not reflecting how life is. So what we did not fully dry our tissue section, but we mounted a cell around it, and we were putting a buffer solution into the cell. So meaning our tissue section is now under physiological conditions. We used ringer acetate because this is a volatile buffer system at pH 8. It is really rather close to physiological conditions. And now we are rastering in this cell over the tissue. What you see here on the right side is the meniscus, and it tends to tear in the red-red zone, but it shows a nice tissue morphology in the bright light microscope. And what you see here, this is how it looks when you look through the microscope, where you try to place and put the nano intender on the proper place, and you are choosing an area to measure your biomechanics. What are the black dots? These are actually bubbles we are sometimes struggling to get rid of. But of course, we are choosing an area with as little bubbles as possible, or we are removing the bubbles when we see them through the microscope. The next step is actually that we are now moving our spherical tip over the tissue, over the meniscus. We are measuring the force maps. That's actually you are measuring the pressure you need to put into the tissue and to release the spherical tip from the tissue. And from that, we are calculating then back the stiffness. So that's a lot of calculation done, because what we are measuring is the tissue on this JPK nanovisor. This is the AFM microscope. And we are moving the tip 50 micrometers further over an area of 1 by 1 millimeter. So we are actually measuring 1, 2, 3, 4, 5, 6, 7 points in the x-axis, and the same number of points the right axis. Every red cross is not only one point where the tip presses into the tissue. The tip itself has only a very small area that is pressing into the material. So we can, per red cross, we are producing a 10 by 10 micrometer force map. So every cross is 10 by 10 micrometer in width and height. Then we are moving the sphere further to the next one, and we are again producing a 10 by 10 micrometer force map. From each point in the force map, we are calculating the stiffness, which is then again visualized in this indentation modulus scheme. And you see these 3 dots, or these 3 points, are overarching approximately only 2 micrometer width in the material, which is approximately 2 collagen fiber thicknesses. This is how it looks like through the microscope. This is the tip you see that is pressing into the tissue, and you are moving always 50 micrometer further over the meniscus material. Of course, then we are generating, and this is not how it really looks like. It looks more like this. This is just a quick sketch over an area of 450 microns. That was the largest area we were measuring. We started with 10 by 10 micrometers. Now we are measuring approximately 450 by 450 micrometer on different areas of the meniscus, and we get information about the stiffness. Of course, we want to know what is the molecular information underneath, so we are moving to mass spec imaging with the very same tissue section. And that means we need to treat our tissue for protein identification. And as most of you know, mass spectrometry and protein identification is based on the digestion of a protein into smaller peptides. And these smaller peptides are measured as accurate as possible by mass spectrometry. And if you have an accurate peptide mass, you can compare it to an in silico digestion and get a probability of correct protein identification. And that is exactly what we are doing, but we are maintaining the lateral information. So how is that done? The tissue section after atomic force microscopy, after biomechanic measurement, is dried in the buffer solution. In the buffer solution. Yeah. Then we wash away the buffer and we run an enzymatic digestion and run our experiment on an ICR instrumentation for accurate molecular masses. And we are generating a peptide map of all the collagens in the tissue section, and we draw the information of where to find these peptides. And that is the outcome. So we find collagen 1, which is not surprising, collagen 2, 3, 4, and 10, for instance. And now we have to correlate this information with the AFM. I'm almost done, Peggy, almost. Okay.
Thank you. Thank you.
I just have to bring the things together now, because now we have the information from one section, from biomechanics and from mass spec imaging. And here you see, this is the area of the whole meniscus. The colorful drawing is the collagen. The red spots are the biomechanic areas where we measured. And what we found is that we also have the stiffest material in the red zones and the softest material in the white zone. And we found out that we have here a very characteristic collagen 1 to collagen 3 ratio, which is actually, which we found for 3 different meniscus regions. So we could confirm what is the reason for the stiffness of a healthy meniscus. Of course, not everything is super easy and straightforward. Here you see that the red-white region is actually not as stiff as the white-white region. And just by mass spec imaging, we found out that we were actually not really measuring in the red-white region, but more in the red-red region, just by looking into the concentration levels of collagen. And with that, I'm almost at the end. This is another example. So we could actually confirm the stiffness, which was found already in former experiments. We correlated also with the fiber orientation, which is our cross-validation. And by that, we are now able to combine biomechanics with protein identification. We try to understand the stiffness and the possibility of meniscal tear to, in the end, better understand MRI images, which are in vivo experiments on that, and patient diagnosis. And with that, I thank my collaborators and you for your attention, and sorry for running a little bit over time.
Thank you so much, Martina. I appreciate that beautiful talk that you gave. Colleen?
Yes. So we have two graduate students from Professor Lingjun Lee's group at the University of Wisconsin-Madison who will be presenting together on imaging quality mass spec imaging and LC-MS for improved confidence in global untargeted mass spectrometry imaging annotation. And so we first have Hannah Miles, who is an F31 recipient working on proteomic investigations into prostate disease for increased understanding of disease pathogenesis, identification of novel biomarkers, and earlier clinical detection. And then we also have Lauren Fields, who is also an F31 recipient and works on improving identification neuropeptides and has developed data-independent acquisition mass spectrometry imaging, and bioinformatics strategies to enable high throughput, optimized identification and quantification of neuropeptides. So take it away, ladies.
Awesome. Go ahead and figure this out really quick.
Lauren, if you could speak up just a little bit, I think, or Hannah, I can't remember.
Yeah, sorry, I was just figuring out the slides. Is everything in just like normal
It looks good.
slideshow view for you guys?
Yes.
Perfect. All right. There we go. All right. Well, I'd like to welcome everyone today and thank the chairs for inviting Lauren and I to talk about our work here today, which is integrating MALDI MSI and LC-MS for improved confidence in global untargeted mass spectrometry imaging annotation. And so I'm going to walk you guys through an example of how we've applied this to our own research, and then I'll hand it off to Lauren, and she'll talk a little bit about the program itself and how you can apply it to your own work in the future. And so, let's see. There we go. So we applied this towards BPH, or benign prostatic hyperplasia, which does affect the majority of the aging male population. It's estimated over 90% of men aged 80 and older will develop this histologically. And that leads to an annual burden of about $4 billion onto the healthcare industry for the management of BPH, and mainly those lower urinary tract symptoms, or LUTs, that drive patients to seek care in the clinic. And there's three main mechanisms of development for BPH. The first is smooth muscle dysfunction, which is clinically treated using alpha blockers. The second is increased proliferation, or that hyperplasia that's in the name, which is clinically targeted using 5-alpha reductase inhibitors, or 5-ARIs. But there is a third mechanism that we study, which is fibrosis via increased collagen deposition and/or density. And that currently has no clinic therapeutic targets. And so that is what we are interested in examining and kind of determining the mechanisms that drive that. But there are some challenges to not only examining fibrosis in the prostate, but just BPH in general. And one of those is the cellular heterogeneity within the prostate itself. And so you can see you have the epithelial and the stromal compartment here. But what we mainly focus on, or try to focus on, is the stromal compartment, because we do know that it contributes to pathophysiologies in prostate disease. What we don't necessarily know is which of the various cell types within the stroma are contributing to disease progression or potentially inhibiting further progression of the disease. And so that spatial information and that localization to certain cell types is necessary when we're examining prostate tissue. But there is an additional layer of difficulty in our analyses, especially is that we want to examine these things in an untargeted fashion. And so typical MSI approaches are done in a more targeted manner, where you have a panel of 10, maybe 20 proteins that you're really focusing in on and examining. But over here on the left, I've kind of demonstrated a short subset of the protein coding genes that we examined related to things like fibrosis, mitochondrial dysfunction, steroidogenesis, aging. This is only a smaller subset, but our total panel is about over 5,000 protein coding genes. And so due to the amount of things that we want to investigate, we need more of an untargeted approach rather than that standard targeted manner that most MSI analyses are done in. And so one way that we tried to opt in to do this is using an on-tissue peptide extraction strategy that was previously published by a postdoc in our lab, Dr. Min Ma, where you have one serial section that undergoes your standard MSI preparation, undergoes an on-tissue tryptic digest, but a serial section also undergoes the same process, and those peptides are extracted from the tissue afterwards and undergo a routine shotgun proteomics analysis to get that MS confirmation for all peptides present within your sample. But there are multiple issues or drawbacks with this strategy. The first is that it's lower throughput, because now instead of generating one data set, you have two separate ones that you have to attempt to integrate back together. And that really slows down this data processing step, especially because a lot of this processing is manual. When we were searching for our specific analyses, we couldn't find one program that would integrate these back together in a more automated manner, or that would analyze them in a combined fashion. They were all kind of separate. One third angle, especially with our research, is a smaller tissue size. When this study was initially done by our postdoc, she was looking at imaging brain tissue in mice. And so you can see here over on the right that the diameter can range anywhere from 8 to 15, around 15 millimeters for tissue diameter. For prostate tissues, however, in mice, the maximum diameter that we have tended to see is around 5 or 6 millimeters. So we need a really high spatial localization and a higher spatial resolution to ensure that we're mapping our peptides back to the appropriate part of the tissue, appropriate cell type, et cetera. And so that is how we came about developing ImSight, which is our software platform that integrates both of these analyses together. And so it starts with an image co-registration, where post-MS imaging data acquisition, you remove your matrix and you can actually do an H&E stain to get that optical resolution and inform your mass spec images to get higher resolution and localization. And then you can integrate your MS results as we've done previously to generate your images. I won't go too much into the details behind this itself, since Lauren will do that in a minute, but I will just kind of briefly overview each of the working parts and then show you an example of that. So I'll start with the image co-registration. Again, this is really crucial to get that accurate spatial localization on our smaller prostate tissues. So you can see down here below, we have our composite mass spec image for our tissue. And then on the top, we have our corresponding optical H&E image of that same section after removing matrix and staining. And so we use the resolution from our H&E image to inform our mass spec image at the end. And so each of those is individually processed before image transformation, where you overlay the mass spec image onto your H&E image. And then this still contains some of those histological dead zones where you may see signal noise in your spectrum, but those are actually gaps within your tissue. And those are removed in your final desired output image, using that to get yourself that better resolution at the end of the day. One thing to keep in mind, however, is that this overlay process, this co-registration, still lacks that MS confirmation that we would get from a standard shotgun proteomics analysis, where you're getting that untargeted profiling. And that's where this next step of the pipeline comes in, where you do that on-tissue peptide extraction and run it through a typical LC-MS analysis. And so with this figure, to note, the top row is just looking at identifications at the peptide level, whereas your bottom row is looking at protein identifications that correspond to those peptides. And so all of our MS results are searched using standard database searching defaults at a 1% false discovery rate. But we did introduce 2 additional filtering metrics onto this before we go through generating our co-registered images. The first is this unique peptide filter, which corresponds to this center column of the figure, where we omit conserved sequences that may map to multiple proteins. For example, collagens is a really good example because many of them share sequence homology or have conserved sequences. And so we want to omit those to ensure that unique peptides that correspond to only one protein are being identified in our final results before we generate images. The second is a filter for peptides with unique mass. And so this is removing those isobaric species that may correspond to two separate proteins, even though they have different sequences, but have that same precursor M over Z. And so you can see we profiled across a variety of different tissue types for all of these figures up here. All of our unique peptides, approximately 90% across tissue type had unique peptides, which was really great to see. And then once we introduce that isobaric peptide filter, we can see a lot more of those peptides were isobaric in nature, and so they are filtered out of our final results, but giving us that higher confidence. And so now that we have those results, we can use them to inform and generate our final overlay images with that increased resolution. And so I'll give you a few examples. The first one is looking at tensin 1, which is essential for myofibroblast differentiation and extracellular matrix or ECM deposition, which we do know to play a role in collagen deposition and then the development of fibrosis. So for this particular protein, we were able to identify 27 unique peptides, 4 of which I've visualized here in the center for you, and then highlighted those corresponding sequences and M over Z values as well in the list on the right. We also took all of our unique peptide images and combined them into one composite image and overlaid that onto our H&E as well, as you can see down here in the lower left, to really get an overall view of that one protein within our tissue. And so we can see a lot of stromal localization, which we would expect for this protein, and we are happy to kind of see in this particular example. But that's not just unique to this one particular protein. We were able to also map it back to 4 other markers of prostate disease. And so these are all shown to play roles in things like bladder incontinence, detrusor instability, which is instability of the bladder muscle itself, fibrosis, and then smooth muscle dysfunction, which you may remember from the beginning is another one of those mechanisms of development for BPH. And so taking all of that into account with IMSIGHT here, all of our analyses were done using formalin fixed paraffin embedded tissues, because in the moving forward, we want to be able to apply this to a larger clinical data sets or those archived clinical cohorts, but these can be used on frozen tissues as well. That image co-registration really helps increase our confidence in our spatial localization with these very small tissues. This process is automated, so it streamlines your analysis, combines it all into one software, rather than using 3 separate ones and attempting to integrate on your own. And then finally, it leads to an untargeted global profiling of peptides on tissue, giving us more of that shotgun proteomics approach spatially that we haven't really had before. And so with that, I'm gonna hand it off to Lauren to break it down a little more for you.
Awesome. Thanks, Hannah, for that wonderful introduction to IMSIGHT and the real power that it has. And so now I want to kind of dive into how IMSIGHT could be used maybe in your own imaging research. So one thing that we recognize as we set out to build this platform based around our own needs is we wanted to be cognizant of the fact that the work that we're doing requires an expertise in microscopy, mass spectrometry imaging, LC-MS, and even bioinformatics. And so that's 4 different areas that most labs might not have a skill set for all 4 of those levels of expertise, or might not have the instrumentation for. And so because of that, it's important to be able to have a program and a software that's able to adapt to the needs of different labs that want to integrate imaging into their research. So for example, we have this in mind. We have, like, for example, a clinician. So clinicians are, skilled and excellent at histological interpretation. And so, for example, maybe they've heard about mass spectrometry imaging and they want to send their tissues off to a core and get them analyzed by mass spectrometry imaging or LC-MS. They get their data back and they want to know what they can do with it. Or you have a lab that's really specialized in mass spectrometry imaging. Maybe they have a little bit of expertise in histological interpretation, but LC-MS is something that they're kind of dabbling in, and they want to see how they can integrate that into their work. Or alternatively, LCMS is the powerhouse of this one lab and they really want to see what MSI can do for them. These are all different scenarios and there's different, spectra in between. And we wanted to build a program that works for all different types of levels of expertise, not just in bioinformatics, but in imaging itself. And so speaking to the bioinformatics side of that, we did some research. So for example, here, this is a paper from 2021 looking at the popularity of different programming languages among scientists. And we see that Python is the most popular language in 2021, and that's a trend that is still popular today in 2024. So we wanted to really make sure that we were keeping up with that trend so that we had the largest possibility of making a program that was applicable to the most people. Additionally, we wrote our program in Python, but applied it in a Jupyter notebook. And this is a method that is very hands-on, kind of guides you through it. And this is a platform that is used in a lot of undergraduate curricula. So this is something that maybe a lot of people are more familiar with than typically, just running something on a command line. And then additionally, the Lee Lab really is proud of having open source software. So for example, here, this is just some of the first lines of ImSight where we show and we kind of walk you through, here's some options and here's how you can apply this to your own data. And so this is kind of what ImSight looks like. So I just wanted to throw this out here before we dive in, just to kind of show how we've thoughtfully tried to prepare this platform for all different levels of expertise. So as Hannah introduced you to the workflow, I'm going to dive a little bit deeper into it. We'll start with H&E, but we'll go through H&E a little bit fast just because this is a really well-established workflow. So we have our FFPE mouse tissue, and of course we do our microscopy. Then we take our original image and we generate that foreground mask from which we can extract out the channel that corresponds to that red pigment. And perform thresholding, and that's where we end up with our smooth Gaussian filter image, and this is the H&E image that we'll use throughout the rest of our workflow to do our localization. Now our MS Imaging workflow computationally is where we had to get a little bit more creative, and so this is where we have, as Hannah mentioned, we do that on tissue digestion, our MS Imaging. Then we do a few different data pre-processing steps to normalize and apply filters, very common steps. And then we do some clustering with PCA followed by t-SNE to do some dimensionality reduction, which then can be overlaid with our clusters over our tissue. And this enables us to identify clusters in an unbiased way that correspond to maybe background signal. And that's what we did here. We removed some of the background signal. And then we're able to build a composite M/Z image, apply a median filter to remove any residual noise, and then interpolate our image to match the dimensions of the H&E image. And so going back to that clustering step, we really harness the power of PCA. It's very fast, very reliable, and robust, but t-SNE is a little bit more precise and sensitive to the global localization. And so that's why we integrated both of these together to really prioritize both speed and global parameters. So here's kind of how I outline this. So we have our H&E next to our clustered image. And here we can see that we've removed clusters 1 and 2, where we've kind of outlined that these are associated with the background. So it's a way that we can quickly highlight and remove pixels that correlate to a certain region of the image, and then we can use that to generate a composite image. From there, we have our pre-processed images, both in H&E and mass spectrometry images. And then we can do our linear transformations followed by our non-linear transformations. So first, looking at the linear transformations, this is where we're overlaying our H&E and our mass spectrometry imaging data. And so we have two kind of options here. Both are using an affine registration, but we have a landmark option and we have an automated option. So for the landmark one, simply you just select up to about 4 pixels that correspond to one another. And then those 2 images will be overlaid just like that. So it's very simple, very easy, but as you can imagine, it's very prone to error, and that can produce variable results, but you do end up with a nice transformed image some of the time. However, we do have an automated option. As Hannah mentioned, all of the steps in ImSight can be fully automated. So this does reveal itself to be unbiased, and it is quantifiable. So we start out with a registered image that's initial. So it's a very poor transformation initially, and then that thing will be automated using the SSD. And so this is computationally expensive, but we found it to perform very well. And then we will do some refinement and some fine tuning on our registration using a nonlinear transformation. And so our big piece here is we wanted to make sure that our transformations were applicable across a wide variety of morphologies of tissues. And so here we see that we started with our H&E and our MSI images. We performed that initial registration, which was then optimized. And then we did our nonlinear transformation. So here we can see that this registration method really works across a wide variety of tissues, which was really something that we were very proud of. Moving on to the LCMS component. So first we did our on-tissue peptide extraction, and then we did our LCMS data acquisition, followed by our data analysis and frag type. You can really use any platform of your choosing. And then as Hannah mentioned, we're able to identify peptides, but we want to filter out those peptides that correspond to more than one peptide, because those can introduce some ambiguity in our results. In the same way, we also filter out isobaric peptides that might have a similar mass, because that can also introduce ambiguity. And then from there, with that knowledge, we have a peptide list from which we can extract the corresponding M/Z images, and then all the images that correspond to a particular protein, which can then be used to build a composite protein image, which can then be overlaid with an H&E image. So as an example of this, here we have cinnamon. And so this was a protein that is an intermediate filament protein, and it does actually have some high expression in some fibrotic diseases. We found this protein to have a high level of expression in our results. So each of these underlined segments represent a peptide that we found. So double underline means that that's just 2 peptides that we found. The blue means that we found it in LCMS, and the red means we found it in both LCMS and MSI. And so with that, we were able to extract the images that corresponded to cinnamon. I put 4 here just for an example, but then of course there were 39 additional ones, which we could then generate a composite image from and then overlay. And so this enables us to begin to understand the localization of cinnamon and monitor it as we progress in our research. So with that, I just want to emphasize the modularity of ImSight. So we've designed this program to be very flexible. There's a number of different platforms that are designed and built in Python for mass spectrometry imaging that don't necessarily encompass this whole pipeline that we built, but they are really optimized for a certain segment. For example, pixel clustering or ROI. And so we wanted to build this platform so it can easily plug and play with these other resources. And so I just wanted to note that it's very flexible in this way. So with that, I just want to note that ImSight is a fully automated platform designed for use in all tissue types. And we support clustering algorithms that might be more effective in different samples. And we were able to accurately identify known markers in prostate diseases. We also fully integrated the use of LC-MS data to establish confidence in a higher throughput manner. And so moving forward, we plan to apply this method to an extended range of tissue. Our next step is to actually apply this to human biopsy samples. And we also plan to examine differential expression of localized markers in mouse models. And so with that, we really want to thank Professors Lingjun Lee and William Rickey for their tireless support of this project and this work, as well as Alexis Adrian and Elliot Petrenitz for their support and help with this. and of course, all of our funding. And we would like to thank Peggy and Colleen for this opportunity.
What a fantastic job, Lauren and Hannah. Thank you so much for that. I think the 3 talks went so well together. So we're going to ask 3 questions, including the audience. Colleen, do you want to moderate that, please?
Yes, so we've got a question from the audience that says, ImSight looks great. Are there steps to remove, adjust, or condense the matrix adducts present in the multispectra that won't be present in the ESI? Oh, Laura, there we go. Thanks.
Yes, so you can take different steps to filter it out in the data processing component. So that is actually done through the database searching platform, so the MS Fragger component. I think that answers maybe the question.
Okay. Martina, I was wondering if your workflow can be used for different species. I know there's a lot of animal models, particularly of cardiovascular disease, that look at some of the elasticity and tension in a tissue.
Yes, of course. It's independent from the biological material. We are just interested in collagen, cross-linking, and tendons and meniscus. So that's the reason why I presented and I've chosen this model. As
It will
long
work
as
across. Go ahead.
Yeah, absolutely. Across every species, across different materials. Yes.
We have another question. I guess this will be the last question from the audience. And this is for Martina. When measuring the stiffness, does the directionality of the fibers have any influence on the measured force? Is there
Yes,
a way that can measure
absolutely.
that?
Absolutely. I mentioned it only quickly because I ran out of time. This is why we are doing quantitative light microscopic analysis as well, QPLM, and where we are looking into the orientation, and we are now correlating that also back to the biomechanics. Yes. Okay, absolutely correct. It's good that you got my talk. Thank you.
I want to actually be sneaky and ask one more question for all of you about ion mobility. So for both Hannah, Lauren, and Martina, is there any opportunities here to use ion mobility in this analysis, maybe pulling apart those isobaric domains?
Yeah, so we've actually started to try and use ion mobility both on the LCMS MS end, but as well as on the MALDI end with the timsTOF. Currently testing it out, but we'll see. So.
Yes, I'm very sure it will help, but we don't have it. And I think bringing the things together, what we have is already really a lot to do. But I'm open that we are sending over sections and happy to see the results.
That's awesome. Lauren, does your pipeline work with, do you think your pipeline will work with ion mobility, and is it vendor neutral?
Yes, so it is vendor neutral. So the imaging aspect uses IMZML files. So I currently do the file conversion and skills, but I imagine that there's other resources that are free to do that. And as far as with the ion mobility, with the imaging, I still need to work out that component, but with the LCMS, that's definitely something that is possible with the because that's why we specifically rely on like MS Fragger, so that we can kind of capitalize on what their advancements are for the LCMS component. So yes, definitely ion mobility will be fully integrated into ImSight.
That's awesome. I look forward to that. So I want to again thank each one of you, Colleen, for helping chair this special session, Professor Marchetti-Deschmann for giving such a great talk on the biomechanics and collagen imaging, and then Hannah, Miles, and Laura Fields for presenting their graduate work, which is, it was a great set of talks. So thank you guys so much.
Thank you.
Thank you for the opportunity, and thanks also for Lauren and you for the nice presentation. It was very interesting.
Oh, thank you.
Thank you.
Yours was very interesting as well. Thank you.
Yeah, thank you.
Thank you. Goodbye.